My name is Leslie Thompson, Leslie-Thompson.
Thank you.
Proceed, please.
Ma'am, can you tell us how it is that you're employed, please?
Yes, I'm employed as a senior criminalist for the Los Angeles County Sheriff's Department, and I am assigned to their crime laboratory.
And where are you assigned?
To the crime laboratory, which is a part of our Scientific Services Bureau.
I mean, what section?
Oh, excuse me, the
Sorry.
forensic biology section.
What is that? The section in the Sheriff's Crime Lab that handles DNA evidence?
That's correct. We
Okay.
handle evidence examination, DNA analysis, and we also make up our crime scene investigation unit.
Okay. And how many years have you been there?
I've been with the Sheriff's Department, it was 16 years last November, and previous to that, I was with the California Department of Justice for approximately 4 years at their Richmond DNA laboratory and their Riverside Crime Laboratory.
Tell us a little bit about your education and training, if you would, that qualifies you for your position as a senior criminalist assigned to the DNA section.
Sure. I have my degree from the University of California at Berkeley in molecular and cellular biology, which is basically biological chemistry. I also, within the Sheriff's Department, completed a 1.5-year-long training program for forensic biology, which consisted of lectures given by senior staff members. I attended courses given by the California Criminalistics Institute. Some of the course titles were DNA extraction and quantitation, STR and DNA analysis, and population genetics and statistics in DNA analysis, to mention a few. My training also included approximately 40 data analysis sets, 20 laboratory experiments, 6 laboratory examinations, 4 written examinations, and 4 oral examinations.
And do you now also teach others in various areas relating to DNA analysis and serology or biology?
Yes, I do.
And approximately how many samples would you say you've processed using various DNA typing techniques over the years?
It's a tough question to answer. I would say I process somewhere close to about 100 cases a year in terms of DNA with the Sheriff's Department. Each case can vary on the amount of samples, from 1 sample to 40, just depending on what that case needs. So, in the tens of thousands.
And have you also qualified and testified before as an expert with respect to DNA analysis and the results?
Yes, I have.
Now, is the Sheriff's Crime Lab accredited?
Yes, we are.
Okay. Are you accredited similarly as the LAPD
Yes,
section?
I believe so.
And do you and the LAPD Crime Lab share space in a building that's located on a particular campus?
Yes, we do.
Okay. And is that at Cal State LA?
It is.
And does the accreditation process require that your lab is audited on a regular basis?
Yes, we are audited every year, not always by our accrediting body. Some years it's by the FBI for their quality assurance standards. Some years it's an internal audit from our quality assurance section. And I believe it's every 4 or 5 years, we are re-accredited through our accrediting body, which is ANAB. And
What does that stand for?
Stands for the American National Standards Institute National Accreditation Board.
And does that accreditation also require that you undergo, along with all of the other analysts in the DNA section, proficiency testing?
Yes, it does.
And what does that entail?
A proficiency test is basically a blind test that mimics casework, and it's created by an outside company. and they send one for each of our DNA analysts or screeners. For DNA analysts, it's every 6 months. And we process that case just like we would an actual case. And then we submit the results to that agency. And it's that agency who checks our results, checks our processes, and ensures that we got the appropriate answers.
Are there also, pursuant to your accreditation, quality assurance measures and controls that are in place within the lab and also specifically within the DNA section?
For our accreditation, but also our laboratory tends to go a little bit above and beyond as well. So there are many quality assurance measures. I could go through some if you would like.
A few.
Sure. So within the laboratory space, we use disposable items to look at the evidence. So a scalpel that you would use one time and then throw away, so as not to contaminate from the scalpel tip. We use sterile swabs. Between each item of evidence, we bleach down our entire area, put up butcher paper. We wear a lab coat, mask, and gloves, and we change those gloves regularly. We examine forensic unknowns or evidence items in a separate space and a separate time from examining reference samples. And a reference sample is a sample from a known individual who may be involved or related to the case. So those are examined completely separately and processed separately to avoid even the sort of possibility of some sort of cross-contamination.
And does your lab also have, just as the LAPD side of the lab has, a computer program that automatically checks for cross-contamination by analysts, that includes analysts' DNA samples within the program?
Yes, we do. And we also have an additional program that checks not just our lab personnel to the sample, but samples to ensure that everything worked appropriately, there was no contamination, and these results are accurate and acceptable and reliable, and we can report them.
And is there also a peer review process in place?
Yes, there is.
So all of your work gets reviewed by a supervisor, the raw data that comes off the instrumentation?
Yes, it does. It actually gets reviewed twice. It gets reviewed by an analyst of equivalent training. So since I'm trained in DNA, it would be an analyst who is also trained in DNA to ensure that we followed our quality assurance written down standard operating procedures, that we performed all the lab work as expected, that we recorded everything, that we were transparent, and that they agree with our results.
Okay.
And then once that review is complete, then the entire file again is reviewed by the supervisor.
And since we've already heard about the basics of what DNA is, I want to ask you about the case specifics regarding laboratory case number under your sheriff's case number 18-036596.
May I refer to my notes, Your Honor? Thank you. Yes, ma'am.
And does that pertain to some evidence that was delivered to the lab under LASD URN number 9-1-8-00363-4016-184?
Yes, that's correct.
And what was the evidence sample that was sent to you for analysis?
I received a toothpaste tube containing an off-white substance.
Is that what we see here on the overhead projector in People's Exhibit 228?
That looks very much like the toothpaste tube I received. However, that's not the photo I took
Yes.
of my evidence item.
Your Honor, I have an additional laboratory photo of what's in 228 along with some results if they could be added to people's 228, 2 additional pages, and in the lower left corner, there's a 1 and a 2 respectively.
Very good.
Showing you this Walters people's 228 with a 1 in the lower left corner. Do you recognize what you see here?
Yes, I do.
And what is this?
This is my photograph of the toothpaste tube that I received as item 2 for this case.
Okay. And is it your pattern that you will photograph the evidence as part of your case package as we see here?
It is.
Okay.
Some items I might not, but in if it's not sort of something you would come across in your normal daily life, I will photograph it, possibly sketch it.
Okay. And with respect to this particular item, did you weigh it as you normally would in an object like this?
I did weigh it. Yes.
And what did that indicate?
This toothpaste tube weighed about 20.5 grams. It's so these are millimeter ticks, so it's really the size of a travel toothpaste tube. And that's sort of significant because, excuse me, on the packaging, the toothpaste tube is labeled as weighing 17 grams. So this was sort of overfilled. It weighed more than what the manufacturer had stated it should weigh.
So the toothpaste had been removed and some other biological fluid or some other type of white, as you said, off-white fluid was inserted into the tube?
There was some other fluid inserted into the tube.
The photograph in People's 228 of the tube stated that the item was passed to the defendant. There was no evidence that the item was passed to the defendant. Is that sufficient, Mr. Booker? Yeah. Okay. Proceed.
Thank you. What did you do with the contents of that toothpaste tube?
I performed some biological testing on that. So I began by taking some of the liquid and I used that to moisten a swab, which is basically a sterile Q-tip. And then I tested that Q-tip. I performed a presumptive test for seminal fluid. It's basically a chemical color change test. So you add certain chemicals, and if seminal fluid may be present, they will turn a purple color. So I performed that test on the swab with the liquid, and it did in fact turn purple. Okay. So that indicated to me that I needed to do more testing because there are some other substances that will give you a positive chemical color change test. So the idea is that if you get a negative, you know that the substance you're testing for is not present, and you can move on to something else. But if you get a positive, now that gives you the indicator to test more definitively for the substance you're looking for.
Let me ask you a question. This color change test, we've heard testimony in this case regarding blood evidence. Is it a similar chemical test to one that would be utilized by experts such as yourself to test for blood? A similar type color change test?
Yes. The exact chemistry is slightly different, but the idea is the same, is that when certain components in the fluid we want to find are present, they will create a visual change in that chemical, and that will indicate that that fluid could potentially be present in whatever the stain it is that you're testing.
So once you got that presumptive positive result for semen, what did you do at that point to confirm?
That led me to create a microscope slide from some of that liquid, stain it. So I added some of that liquid to a glass slide, I allowed it to dry, I added certain stains that would color the different cell types, and then I visually looked for sperm cells using a microscope within that liquid. And I did, in fact, find a large amount of sperm cells within that liquid.
Is there some type of rating system that you use based on the volume of sperm cells?
There is a grading system. It's not very defined, but if you can find a multitude of sperm cells in each area, because you aren't able to look at the entire stain in one shot, you have to move the slide around to view the entire stain. So in each viewing, if you can see a multitude of sperm cells, we would rate that as a 4+, which is the highest. And then 3+ would be a little bit less, and maybe not in every viewing, that's a 3+, and a 2+ would be less, and in some viewings, and so on, as it goes down. So this particular side rated as a 4+.
And once you looked at it under the microscope, what is the next step that you did?
So the next step is that I wanted to perform DNA analysis on the fluid with the sperm cells in order to obtain the DNA profile from that sample.
And so subsequently, did you do that so that you could proceed through to the DNA process?
Yes, I did.
Okay. Also, subsequent to that time, did you receive a reference sample for an individual by the name of Robert Baker? When we say reference sample, again, we're talking about a known sample.
Correct, and yes, I did.
And did you receive that from Randy Zepeda, who works in the DNA section of the LAPD's crime lab?
Yes, I did.
And is he someone you've known for years from your work in the lab?
Yes, he is.
Bless you.
He might have even been in one of my classes.
Okay. And what was the form of the sample that you received from Mr. Zepeda?
It was basically a sterile swab as well. It's called a Bodie swab, and Bodie is just the name of the company that makes these swabs.
It's a buccal swab, correct?
Correct.
And what is a buccal swab?
A buccal swab, it's simply, a buccal is a epithelial or non-sperm cell. It's called a buccal or buccal. And those line the inside of your cheeks. And so in order to collect this type of a reference sample from a swab, you would put it in your mouth and rub it on the inside of your cheeks on both sides. And in doing that, the swab would absorb cells from the inside of, from the inside of your mouth.
Now we've heard about the some of the steps in the DNA testing process. What testing kit did you employ in this case?
Our lab has Fusion 6C validated for use.
Okay. And is that the same testing kit that's utilized on the other side of the lab by the LAPD?
Yes, it is.
Okay. So you went through the same steps that they would go through, including the extraction, the quantitation, amplification, and then the analysis and detection, correct?
That's correct.
However,
Exactly. However, I don't know what types of samples that were looked at LAPD, but there's a specific type of extraction that's just for when you have a sample where you suspect there are sperm cells present. And LAPD has the same type of extraction, but I don't know if they performed, it's called a differential extraction. I don't know if they performed a differential on any of their samples. I don't know their case. But because I identified sperm cells in this sample, I did perform a differential extraction.
okay. And what does the differential extraction process do?
So ultimately what it does is it's able to separate sperm cells from non-sperm cells. So it takes this sample, and in this case, I put some of this liquid on a swab and put the swab in a very small sterile tube to begin DNA analysis. So the way the differential extraction works is I added specific chemicals that would only break open the non-sperm cells, allowing the non-sperm cell DNA to be released into solution while the sperm cells still stayed intact. So a sperm cell is a little bit different from a normal cell because it's designed specifically to travel through a hostile environment and then deliver just its DNA to an egg. So it's in sort of it's in a capsule, and that capsule is reinforced with sulfur bonds. It's just reinforced, so it's a little bit stronger than a normal cell. And we take advantage of that, and we're able to add those chemicals that will burst open the normal cells, but leave those sperm cells intact. Once we do that, we're able to take the liquid that has the non-sperm cell DNA away from the sperm cells. And now we have two samples. We have a sample that consists of non-sperm cell DNA and a sample that consists of sperm cells that now we can add special chemicals to that will allow us to break the sperm cell capsule open and release its DNA. So we've created two different fractions or portions or samples from the one. And it's not exact. It's more of we're concentrating in this one sample the non-sperm cell DNA, and we're concentrating in the second sample the sperm cell DNA.
And that's the differential extraction is a standard process utilized by crime labs around the world. In order to separate out, if you have a sample that you suspect contains semen, sperm cells, from the epithelial or the non-sperm cell fraction, correct?
That's correct.
All
And
right.
it's used because normally when you have a swab and you're looking for the semen, it's from a part of someone's body, and usually a mucous membrane, like the inside of your mouth or the vaginal canal. And those mucous membranes produce a lot of cells throughout the day. And so those swabs would contain a lot of non-sperm cells. And so that could potentially overwhelm the sperm cell DNA profile. So we have a better chance of seeing everything that's there if we're able to separate the two and then perform DNA on each one individually.
Okay. So, did you issue in this case a DNA report regarding what you did, your analysis, and the results you obtained that was dated September 15 of 2018?
Yes, I did.
And did you develop profiles, a reference profile for this individual, Robert Baker, based on the evidence you were provided by Randy Zepeda?
Yes, I did.
And did you also develop evidence profiles, DNA evidence profiles for the sperm and non-sperm fraction from the liquid contained inside of this toothpaste tube?
Yes, I did.
Okay. And did you make a comparison between the reference profile belonging to Robert Baker to the unknown evidence profiles that you developed using that differential extraction?
Yes, I did.
And tell us your results, please. Let's start with the epithelial fraction or the non-sperm cell fraction.
Absolutely. So for the sample of the semen from the toothpaste tube, the non-sperm cell fraction, I recovered a DNA profile consistent with coming from one individual. So it was a single source profile, and Robert Baker was included within that profile. I also performed some statistical analysis which will give meaning or weight to that inclusion, if I may.
Yes.
I performed what's called a likelihood ratio. And a likelihood ratio, it compares two separate or varying ideas.
Two
One,
hypotheses, basically competing hypotheses?
two competing hypotheses, exactly. So one is that given this DNA profile we recovered from the evidence and this DNA profile from this individual, how likely is it that this profile from the individual could have made up the profile that we recovered from the unknown versus I have this DNA profile from my unknown and a DNA profile from anybody else in the world, any profile I want. How likely is it that that could have made up this DNA profile? And when you compare the two, If it's more likely that the individual you're looking at could have made this DNA profile, then your likelihood ratio is going to be larger. If it's not very likely, meaning that the genetic profile really doesn't match or it's not consistent with the deconvolution of your unknown,
The interpretation, basically, of your unknown sample.
basically, the interpretation of your unknown sample, then that would be an exclusion. And so in this case,
You said that Robert Baker was included.
that's correct. And the DNA profile is approximately 9 times 10 to the 28 times more likely if it originated from Robert Baker than if it originated from an unknown individual. So in terms of what that number means, within our laboratory at the time, any likelihood ratio greater than 1,000 is an inclusion. Any likelihood ratio between 0.001 and 1,000 is inconclusive. Anything less than 0.001 is an exclusion. That means this individual could not be the contributor of the DNA. So 1,000 is a 1 with 3 zeros, and that would give you an inclusion. This is a 9 with 28 zeros. It's a huge number. It's not even just thousands of thousands, it's millions of millions. So it's very strong probability that, in fact, Robert Baker was the contributor of the DNA that we found in this profile.
So once you make that determination, did you move on to the analysis of the sperm fraction of the contents of that toothpaste?
Yes, I did.
And what was the result?
I found from the sperm fraction of a sample of semen from the toothpaste tube a DNA profile consistent with coming from one individual, so a single source profile, and Robert Baker was included within that profile. I performed the same type of statistical analysis, and the DNA profile from the sperm fraction of the sample of semen from the toothpaste tube is approximately 9 times 10 to the 28 times more likely if it originated from Robert Baker than if it originated from an unknown individual.
So you basically got the same statistic from your likelihood ratio calculation.
Yes.
And would that be what you would expect, given that you're talking about a single source sample?
Yes, and given that in looking aside at just the profiles, they were both single source, they were both male, and they both contained the same alleles.
Single source meaning came from one person as opposed to a mixed sample, like if you're talking about, when you mentioned the differential extraction, if you were talking about an item that was recovered, let's say, from a sexual assault kit that might have the victim's DNA along with a perpetrator's DNA.
Correct.
Okay. Thank you. I have nothing further.
That's an important.
Thank you, and I'll never look at appropriation the same again this time.
Glad I could help.
We have no questions.
Thank you, ma'am. Your excuse.
Thank you, sir. Thank you, Your Honor.
Do you make other witness?